Using carbon isotope. Before we begin, let's recap on the structure and function of phloem. Pause the video to think. That's right, the phloem is made out of the C-cube element and the companion cell. Do you remember what is the structure and function of xylem? Let's do a comparison. Xylem, as you know it, is dead, whereas phloem is alive due to its companion cells. The transport of water and mineral salts in the xylem vessel is unidirectional, whereas the transport of manufactured food in the phloem is bidirectional. The transport of manufactured food substances such as amino acids and sucrose in the plant is known as translocation. How did scientists find out that manufactured food substances are transported in the phloem? Before we get to that question, let's recap on the where and how food is being manufactured in the plant. In nutrients in plants, we learned that food or glucose is made in the leaves by chlorophyll during photosynthesis. Carbon dioxide is used to make glucose. The chemical formula of glucose is your C6H12O6. Glucose is then used to make sucrose. Why do we have to convert glucose to sucrose? Write down your answers in the worksheet. Sucrose is then transported through the plant. Where is it transported and how is it transported? We now know that it is through the phloem. However, how did scientists in the 20th century know that it was transported in the phloem? That is through radioactive labeling. The first step is to supply radioactive carbon to an intact leaf enclosed in a sealed chamber. Instead of using the normal carbon-12 for its molecular weight, carbon-14 is used, which is radioactive. Radioactive carbon became popular after the World War II, available to researchers. Radioactive glucose is then made using the radioactive carbon dioxide, and sucrose is also being made from the glucose and transported through the plant. As the molecules that are radioactive can be traced, it became possible to track the flow of these molecules through the cells. This can be done by cutting a section of the stem and exposing it onto an X-ray photographic film. Predict the regions where radioactivity would be detected. Draw out your observations and explain why. That is the end of the video. Please fill in your worksheet and explain your findings to your group mates.